Good afternoon. I'm Sir Franz, and this time we will be discussing the targets of drug action. And our goal is by understanding the general nature of receptors, we can focus our search for new drugs. And by understanding the general nature of receptors, we can be able to correlate our observations when testing pharmacologic actions of new drug substances with biologic systems. So, our general objective would be to discuss the different types of receptors. The first type of receptor are receptors that affect concentrations of endogenous ligands. These receptors will alter the synthesis, storage, release, transport, or metabolism of endogenous substances, and thereby affect the levels of these endogenous substances. We are talking about neurotransmitters, hormones, and other intracellular mediators. What is shown in this slide is an illustration of the noradrenergic synapse and the different um, drugs that affect the levels of noradrenaline. Drugs acting here, such as alpha methyltyrosine or metyrosine, inhibits the synthesis of norepinephrine and thereby decreases the levels of norepinephrine. Another drug that I like to highlight here is cocaine, which blocks the reuptake of norepinephrine and thereby increases the levels of norepinephrine available in the synapse. And lastly, amphetamines, which will promote the release of norepinephrine into the synapse. Thereby, by altering the levels of norepinephrine in the synapse, we can pharmacologically manipulate the magnitude of effect of norepinephrine in the system. Other examples of neurotransmitters that are affected in their levels by receptors include the cholinergic and the serotonergic synapses. So we're talking about the levels of acetylcholine and the levels of serotonin and their associated effects. The next kind of receptor would be receptors that regulate the ionic milieu. By this, we are referring to the concentrations of ions that are found in the blood, the urine, and the gastrointestinal tract. So for us to understand that, we should first define that an ion is any element with a charge or a valence that is not zero. So we should also understand that ions affect the physiologic function of cells and the body in general by altering the voltage and membrane potentials of cells. So receptors that regulate the ionic milieu include ion pumps and transporters. When we refer to pumps, we are describing biologic systems that transfer ions from a gradient that is low into an area where there is high concentration of these ions. So we are going against the concentration gradients. And transporters are proteins that facilitate diffusion of these ions, and therefore we are talking of going with the concentration gradient, just facilitating the efficiency of that gradient using proteins. These transporters are especially expressed in highly developed epithelial cells. So here in your photo is an illustration of these ionic regulators in action. So here is the nephron, and most of the diuretics affect ion transporters in the nephron. And for at now, you should be able to realize that the nephron is basically a specialized type of epithelium. For example, the thiazide diuretics affect the levels of sodium and chloride in the distal convoluted tubule, where these transporters are expressed by epithelial cells. So, if you will direct your attention on the upper right portion of the image, you will see the site of action of the thiazide diuretics. And the protein, the receptor that we are focusing our discussion on would be the symporter for sodium and chloride. By the term symporter, we mean that this protein catalyzes the transport of sodium and chloride together as they enter the intracellular compartment. So, by symport of sodium and chloride, the levels of sodium and chloride in the urine decreases. So, in that way, this transporter affects the ionic concentration of sodium and chloride in the urine. Okay, another example are loop diuretics that affect the sodium potassium dichloride co-transporter in the loop of Henle. Okay, so if you will direct your attention on the left lower portion of the image, you will see the site of action of loop diuretics. And similarly, this is a symporter for three ions, sodium, potassium, and chloride. You will also notice that potassium is reabsorbed by keeping the levels of potassium intracellularly low. So this is um, done by the efflux of potassium. So whenever the intracellular levels of potassium is low, then the concentration gradient is established such that in the urine there is high level of potassium and in intracellularly there is low level of potassium. Okay. So in that way, these proteins also alter the levels of sodium, chloride, and potassium in the urine. And lastly, aldosterone and aldosterone antagonists alter the expression of ion transporters at the level of the gene. So if you will direct your attention at the right lower quadrant, you will see where these aldosterone antagonists function. Okay. So examples of ionic regulators in the form of receptors are also found in the alimentary tract or the gastrointestinal tract. And an example of that would be the proton pump, which is found in the parietal cells of the stomach. And by targeting the proton pump using proton pump inhibitors, we are knocking down their activity. The levels of acid released by the stomach goes down by up to 90%. And this inhibition is irreversible, which means that the parietal cell, in order to re-establish its secretion of acid, would have to generate by protein synthesis new sets of proton pumps. Okay. The next type of receptors that we'll be talking about are G protein coupled receptors. And this is how, in general, the G protein coupled receptors function. First, they are unbound by the ligand, and that is their natural state. And in that unbound state, the receptor and the G protein heterotrimer form a complex in the membrane with the alpha subunit bound to GDP or guanosine diphosphate. So let's try to analyze that. First, let's highlight that the G protein coupled receptor, the complex, is found in the cell membrane. So that's the first key takeaway, that they are found in the cell membrane. The second one is that when it is inactive, it occurs as the unbound state. So the second takeaway, the inactive is the unbound state. Okay. Third, in that state, they form a heterotrimer. So that means that bound to the receptor are three different proteins that form a complex: the alpha, the beta, and the gamma complex. Okay, or G proteins. Okay, so what's special is that the alpha subunit is bound by guanosine diphosphate, which is a low energy transducer. Okay, so let's now go into when binding happens. When that happens, there is a conformational change of the complex, and you will see that this change in shape results to the disaggregation of the three proteins, the alpha, the beta, and the gamma. The alpha subunit separates, and this alpha subunit, instead of being bound by guanosine diphosphate, is now bound with guanosine triphosphate, which is a high energy transducer and therefore activates the alpha subunit. Another product of that dissociation is the formation of the beta gamma dimer. So these two, the alpha subunit, which is bound by GTP, and the beta gamma subunit dimer interact with intracellular receptors also, or intracellular proteins, which will mediate the functions. So we will have a separate slide for this one. You will see at the middle of this image that there is a cycle between the activated and the inactivated or deactivated state of the alpha subunit. And that is observed by binding of either GDP or GTP. So that one point of control of this system is by hydrolysis of GTP. By releasing that one phosphate, the alpha subunit becomes deactivated as that GTP becomes GDP. And that deactivated alpha subunit couples again with the beta and the gamma subunits to form the basal state of the complex. Okay, so let's now go into how these subunits transduce their effects. Okay, and that is the concept of effector modulation. The G protein subunits interact with other intracellular proteins that mediate the translocation or activation of functional proteins. So let's try to understand that one. The alpha and the beta gamma dimer will interact with other proteins floating in the cytoplasm. And these proteins, intracellular proteins, will then be the ones to phosphorylate, dephosphorylate, or move intracellular proteins into their proper locations, whether these are regions of the cell, organelles, particular portions of the cell, such as near the nucleus or in the cell membrane. So by activating these intracellular proteins, the subunits are then able to transduce their signals, which were originally given off by the ligand. Okay, so parang may cascading of information. Una sa ligand, and then sa receptor. And then yung information from the receptor will be transmitted via conformational change into the G proteins. The G proteins will then dissociate and from there act as activating proteins also, which will activate cellular proteins, which will then activate further other proteins, which will then be the ones to give off the function. So one concept here is the concept of magnification or amplification. Because one activated intracellular protein will interact with multiple subsequent targets, the signal is amplified. So if you can understand that one, that there is only one ligand that binds to the receptor, and downstream there will be multiple effects. That is an illustration of amplification. So if you will take a quick look at this image, in the middle is the phosphorylation and the conformational change of membrane proteins. Okay, so let's first take a look at the right portion of this image, and this is the effector modulation produced by the alpha guanosine triphosphate complex. So the alpha subunit bound by GTP. It couples with an effector protein, and these effector proteins can either be structural proteins or enzymes. And examples of these enzymes are adenylyl cyclase, AC, and the increase in the activity of adenylyl cyclase is produced by G protein subtype S or GS. And when the levels of or the activity of adenylyl cyclase increases, then the levels of cyclic adenosine monophosphate will increase, and cyclic adenosine monophosphate will activate protein kinase A, and that protein kinase A will further phosphorylate other proteins, activating the entire cell. And an example of a ligand that goes through that pathway is epinephrine when it binds to the adrenergic receptor, usually the beta receptors. Okay. On the left side of this image are mediation by the beta gamma dimer. So similarly, the beta gamma dimer will bind with the effector protein, and that effector protein can either be enzymes such as inositide triphosphate activated kinase PI3K, or these dimers will translocate into the cell membrane, the potassium channel, which will cause the efflux of potassium intracellularly into the outside environment, or it will translocate the calcium channel, which will cause the influx of calcium, thereby increasing the intracellular concentration of calcium and activating various enzymes that rely on calcium, such as enzymes of the Krebs cycle and glycolysis. So in that way, calcium activates the entire cell metabolically. Lastly, we will be talking of nuclear receptors. And by the name itself, these receptors are found floating in the nucleoplasm or integrated into portions of the gene. And thereby, they will have to be activated by ligands that can traverse the cell membrane into the cytoplasm and enter the interior of the nucleus. These nuclear receptors function as transcription factors, and the effects that they produce can be seen over hours to months. That means it is long term. They regulate processes, complex processes such as reproduction, development, and metabolism of the organism. And the ligands that bind these receptors are usually circulating steroid hormones. So what is seen in this image is how the nuclear receptor functions. So the nuclear hormone receptor, which is illustrated here as OR, is shown in a complex with the retinoic acid receptor, RXR, whose ligand is vitamin A. RXR, the blue one. When an agonist, which is manifested here as the yellow triangle, and the co-activator bind, a conformational change in the complex, particularly in helix 12, which is the black bar, occurs. And when this happens, gene transcription is stimulated. When the transcription is facilitated, transcripts in the form of mRNA will increase in concentration in the cytoplasm. And when these mRNA transcripts are translated into proteins, the levels of proteins also increase. Okay, so examples of hormones that can go through this pathway are the steroidal hormones, such as estrogen, the androgens, glucocorticoids, and mineralocorticoids. An exception to that is the non-steroidal hormone, thyroid hormone, which is a peptide hormone but acts through nuclear receptors. So to take that example further, when glucocorticoids bind to their receptors, they will facilitate the transcription of the glucocorticoid response element, GRE, whose products are enzymes that take part in metabolism such as glycolysis, enzymes of the Krebs cycle, and even enzymes or protein products that depress the immune system by blocking the transcription of um, products that respond to cytokines such as interleukin 1 and tumor necrosis factor. So in this way, by blocking the transcription of the products of interleukin 1 and TNF, then glucocorticoids such as cortisol are able to inhibit the immune response. Okay, and thus find their way into applications such as um, immune suppression in the treatment of rheumatologic disease or the treatment of severe infections such as sepsis, where the immune system is dysregulated and harm the host. So that's it, and this is my reference for this lecture. Thank you for listening.